Welcome to this GCSE Biology revision video on plant hormones. Plants are highly responsive to their environment, even without brains or nervous systems. This video will explore how they detect and respond to stimuli and the key hormones involved. We will cover 5 main sections: plant responses, auxins, other key hormones like gibberellins and ethene, agricultural applications, and exam tips. This structure will guide you through all the essential topics. Chapter 1 focuses on how plants respond to their environment. Like all living things, plants must respond to changes to survive. They react to stimuli like light, gravity, and water to maximize photosynthesis, anchor themselves, and absorb water. Since plants are rooted in place, they can't move away from danger or toward resources. Their solution is to use chemical signals called hormones. These hormones coordinate responses by being produced in one part of the plant and triggering changes elsewhere. Plant hormones are chemical messengers produced in small quantities that regulate growth and development. They are synthesized in specific parts of the plant and transported to target cell trigger responses like cell elongation or fruit ripening. Key GCSE hormones are auxins, gibberellins, and ethene. Chapter 2 is about auxins, the key growth hormone. Auxins are produced at the tips of shoots and roots and control the direction and rate of plant growth. The most common auxin is indole acetic acid, or IAA. Auxins control growth through 3 main steps. First, they are produced at the tip. Second, they are transported downwards, moving sideways in response to light or gravity. Third, they stimulate cell elongation, causing the plant to bend where concentration is high. A crucial principle to remember is that auxins cause cells to elongate, meaning more auxin equals more growth. This is the single most important principle behind both phototropism and gravitropism. Chapter 3 covers phototropism, which is growth in response to the direction of light. This allows plants to maximize light for photosynthesis. The word comes from the Greek photo for light and tropos for turning. Shoots are positively phototropic, meaning they grow toward the stimulus, which is light. This positions leaves to absorb maximum sunlight for photosynthesis. Roots, however, generally don't respond strongly to light and primarily respond to gravity. The mechanism of phototropism involves 4 stages: light strikes, auxins shift, shaded cells elongate, and the shoot bends. This auxin redistribution drives the bending motion, a key mechanism to understand for your exam. In phototropism, auxins move away from light and accumulate on the shaded side. This results in a lower auxin concentration on the light side, causing less elongation. The higher auxin on the shaded side causes faster growth, resulting in the shoot curving towards the light source. Directional light triggers the lateral movement of auxins within the shoot tip. These auxins migrate to the shaded side, which stimulates more cell elongation there. The shoot then curves towards the light, demonstrating a positive phototropic response. Chapter 4 introduces gravitropism, also known as geotropism, which is a plant's growth response to gravity. This response ensures roots grow downwards into the soil toward water and minerals, and shoots grow upwards toward the light. Roots are positively gravitropic, growing with gravity downwards. This helps anchor the plant and absorb essential resources. Shoots are negatively gravitropic, growing against gravity upwards, ensuring leaves are in the best position for photosynthesis. Remember, positive means with gravity, negative means against gravity. Auxins control gravitropism, but they have opposite effects in roots and shoots. In shoots, high auxin means more growth. In roots, high auxin actually means less growth. The same hormone, but 2 different outcomes. Looking closer at roots, when a root is horizontal, gravity causes auxins to accumulate on the lower side. Critically, high auxin concentrations inhibit cell elongation in roots. This causes the lower side to grow more slowly, curving the root downwards, a positive gravitropic response. This table summarizes the opposing effects of auxins in shoots and roots. This contrast, where the same hormone leads to opposite growth results, is a frequent topic in exam questions, so make sure you understand it. Chapter 5 shifts to gibberellins, another group of plant hormones. They are produced throughout the plant, including in seeds, leaves, and roots, and have important roles in seed, growth, and flowering. Gibberellins have 3 main roles: stimulating seed germination by activating enzymes, promoting stem elongation leading to taller plants, and initiating flowering, often in response to environmental cues like day length. In agriculture, gibberellins are used to produce larger seedless grapes. They are also used in the brewing industry to speed up barley germination for malting and to control plant height for different crop requirements. Chapter 6 introduces ethene, the unique gas hormone. Ethene is naturally produced by ripening fruits and is critical in controlling the ripening process. Its gas form makes its effects easily spreadable and exploitable in the food industry. Ethene works through a positive feedback loop. One ripe fruit produces ethene, activating enzymes, causing cell softening in neighboring fruits. This leads to more ethene production and further ripening, which is why fruit can spoil so quickly. In practice, ethene allows for controlled ripening. Fruits like bananas are picked green, shipped, and then exposed to ethene gas at the destination to trigger ripening just before sale. Conversely, storing fruit in low ethene environments slows ripening and extends shelf life. Chapter 7 focuses on how modern agriculture utilizes plant hormones, often in synthetic form, to boost productivity, manage growth, and reduce crop losses. These applications are central to food production today. Rooting powder contains synthetic auxins. Dipping a cutting in it stimulates roots to grow from the stem end. This technique allows commercial growers to quickly clone valuable plants, producing genetically identical stock reliably. Selective weed killers use synthetic auxins to cause uncontrolled, abnormal growth in target plants, effectively killing them. They are selective because they primarily affect broad-leaved weeds, leaving narrow-leaved crops like cereals unharmed. To summarize agricultural uses, auxins are used in rooting powder and selective weed killers. Gibberellins aid in larger fruit production and germination. Ethene is key for controlled ripening in food transport and storage. Here is a comparison of the 3 key hormones for exam focus. Memorize their production sites, main roles, and commercial applications to ensure you can distinguish between them clearly in your answers. Finally, here are some key exam tips to help you avoid common mistakes. Mastering these points can maximize your marks in questions about plant hormones. The first exam tip is the auxin trap. Never say auxins are attracted to light. The correct answer is that auxins move away from the light to the shaded side, causing greater cell elongation there, which bends the shoot toward the light. Exam tip number 2 focuses on the opposite effects in roots and shoots. Remember, in shoots, high auxin promotes growth, but in roots, high auxin inhibits cell elongation. You must mention this difference when explaining why shoots grow up and roots grow down. Exam tip 3 is about defining your terms. Ensure you can clearly define phototropism, gravitropism, hormone, and cell elongation. For example, phototropism is growth in response to light, and a hormone is a chemical messenger. Let's do a quick-fire revision quiz. Test your knowledge. Which side of a shoot has more auxin when light shines from one side? Are roots positively or negatively gravitropic? What's gibberellin's role in seeds? Why does one rotten apple ripen others? The answers are below. You've got this. We've covered the key mechanisms of auxins, the effects of gibberellins in germination and growth, and ethene's role in ripening. Keep these concepts and applications clear for success in your exam. Good luck!